Hi, this is Kristen, and I am going to do a quick exam review. I wanted to focus on the cardiovascular system and begin with specifically the blood vessels. This is in preparation for class exam 2 in anatomy and physiology, biology 231. Okay, in preparing for the exam, it is important that you be familiar with the different layers of the blood vessel wall. So that would include, whoops, seem to be having some trouble showing the PowerPoint right now. One moment, please. Okay, all right, here we go. So the three layers are the tunica intima, which is an intimate contact with the blood, and it is smooth and reduces friction. It's important to recognize that the tunica media, which is the thickest middle layer, is mostly smooth muscle, also includes some elastin, connective tissue, but it's most important for uh, causing vasoconstriction and vasodilation. So tunica media is an important layer. The tunica externa, also known as the tunica adventitia, anchors the vessel to the surrounding structures and is thinner than the tunica media. It's also important to be able to understand that the tunica media is innervated by only sympathetic vasomotor nerve fibers, which means that the vasodilation and vasoconstriction is only controlled by the fight or flight branch of our autonomic nervous system. And so when the impulses speed up, that will cause vasoconstriction, and when there's a decrease in the rate of sympathetic stimulation of the blood vessels, then vasodilation will occur. Okay, it's important to understand that there are arteries that carry blood away from the heart, veins that return blood to the heart. The capillaries are microscopic blood vessels that increase dramatically in surface area. And when blood reaches the capillaries, even because the surface area of the capillaries increases so much, the flow of the blood decreases, and capillaries are under very low pressure. Capillaries are what are responsible primarily for the exchange of nutrients, wastes, and respiratory gases with the cells. So they're sometimes called exchange vessels. Now, arteries can be divided into three categories. The elastic or conducting arteries, which have a great deal of elastin in them. Their primary function is to smooth out pressure fluctuations. And the largest artery is the aorta, a perfect example of a conducting or elastic artery. Then most of the arteries in the body are named according to their location, and those are primarily the second largest diameter arteries, downstream from the conducting arteries, and they're known as the muscular or distributing arteries. And then finally, we reach the arterioles. Now, the arterioles are primarily involved in vasoconstriction and vasodilation. They're the smallest arteries right before the capillary beds, and they control the amount of blood that enters the capillary beds. If we go on to look at looking at one specific picture, this one, you can see that the greatest change in blood pressure occurs in the arterioles. So we have the highest pressure in the aorta, and then pressure slowly declines as blood moves away from the heart. And blood pressure is shown on the y-axis. The categories of vessels are shown on the x-axis. And you see the steepest part of this blue line occurring at the arterioles, indicating that there's the largest change in pressure here. And so here, these arterioles are capable of vasoconstricting, which will increase the resistance to blood flow and slow down the blood as it is distributed to several microscopic capillaries. Okay. It's also important to be able to calculate mean arterial pressure. You should just have that memorized and understand that mean arterial pressure it's basically the average blood pressure in the body. Our heart spends more time in diastole than it does in systole, so you'll notice that the mean pressure is closer to the diastolic blood pressure in this graph. It's closer to 80 than it is to 120. And so there's the mean arterial pressure right there. It's MAP, or mean arterial pressure, is the diastolic blood pressure plus, and then pulse pressure divided by 3. Pulse pressure is systolic blood pressure minus diastolic blood pressure. So that's something that you should have memorized. Okay. Another important point to keep in mind is that let's see. Another important point. Oops. That's weird. How did that happen? Somehow I switched over to, so strange. Hold on. Sorry about that. One moment. I want to look at the blood vessels. Another important point is that there are also venules and veins, and venules are the smallest veins. Veins are the larger diameter blood vessels that return blood to the heart. It's important that you understand arteries are under high pressure, veins are under quite low pressure, and they, at any given time, contain the most blood in the body. They are considered to be capacitance vessels. And so you'll notice in this image that 65% of the blood at any given moment can be found in the veins. Veins, because they're under low pressure, have to have one-way valves in order to return the blood to the heart. Okay. It's also important that you have a pretty good understanding of the different types of blood vessels that are found in the body. So, we have 3 different types of blood vessels. Capillary structure is shown in this image of the continuous capillary. Continuous capillaries are the most common and the least permeable, which means that fluid and small solutes are least likely to seep out of the capillaries into the interstitial space with these continuous capillaries. The fenestrated capillaries have a little window called a fenestration, or a pore, and this type of capillary will be found any place where there is going to be a lot of filtration, such as the kidneys, or even reabsorption, such as the intestines. The leakiest and most permeable capillary is called the sinusoid capillary. And this is located in the liver, bone marrow, spleen, and in the adrenal medulla, which is a hormone-secreting gland. Okay. Another thing that's important to remember is that the actual true capillaries that are exchanging substances with the cells in the interstitial fluid are different than the vascular shunt. In this image, the vascular shunt has little pre-capillary sphincters, which are shown here, and they're made up of smooth muscle, and when they constrict, they can prevent blood from entering the true capillaries. And the true capillaries, again, are the things that exchange substances with the cells. Okay. Another thing that's important to note is that blood is returned to the heart through breathing, the changes in pressure in the thorax from breathing, from contraction and relaxation of skeletal muscles in the legs, in conjunction with the one-way valves, and also some vasoconstriction of the veins. There is some smooth muscle in the walls, however, not nearly as much as in the arteries. Okay, so in order for blood to flow, you have to have a difference in pressure called the pressure gradient. So there's a significant difference in pressure between the aorta and the superior and inferior vena cava. And in a healthy person, that ranges from about 120 millimeters of mercury to approximately 1 or 0 millimeters of mercury in the superior and inferior vena cava. An important relationship to understand is that peripheral resistance, or resistance to blood flow in the systemic circulation, is caused by an increase in blood viscosity, which is normally constant, the length of the blood vessels, which is normally constant. The thicker the blood, or the more viscous the blood, the higher the resistance. The thicker the blood vessel, or the longer the blood vessel length, the greater the resistance to blood flow. But the most important component would be the blood vessel diameter. If there is a change in blood vessel diameter, it will have the largest impact on resistance. So, if you decrease the radius or diameter of a blood vessel through vasoconstriction, it will greatly increase the resistance to blood flow. On the other hand, if you vasodilate, it decreases the resistance to blood flow. So it's important to understand the relationship that blood flow, which is the same thing as cardiac output, is directly proportional to this pressure gradient. In other words, the difference in blood pressure between the aorta and the superior and inferior vena cava. As you increase the difference in pressure between the aorta and the superior and inferior vena cava that return blood to the right atrium, you increase the blood flow or cardiac output. If you increase resistance to blood flow, you decrease blood flow. If you increase resistance to blood flow, you decrease cardiac output, since cardiac output is really blood flow. So this relationship, I think, shown here, it's very important to understand that. Okay. As you decrease resistance or vasodilate, you are going to increase blood flow or cardiac output. So because flow and resistance are inversely related, as one goes up, the other goes down. Resistance, as you can see, is also inversely related to the pressure gradient, or delta P. Okay. All right. It's also important to understand that as blood leaves the aorta, the velocity of the blood flow at the aorta is quite fast because the heart is ejecting that blood into the aorta under high pressure. However, as the blood leaves the aorta and travels through the muscular arteries toward the arterioles, you'll notice that the speed of the blood dramatically decreases. Well, this is largely because the surface area, the total surface area of the vascular bed greatly increases when we reach the capillaries. In fact, the greatest surface area of the blood vessels occurs in the capillaries, and you'll notice that's where we have the slowest speed of blood. And that's important because it gives the blood and the cells time to exchange substances. Okay. Another thing that's important to remember is that it's important to understand that hydrostatic pressure is the force of a fluid against the membrane containing it. That colloid osmotic pressure is an opposite force. It's an opposite to hydrostatic pressure in that these large non-diffusible molecules, since they can't move through the membrane, they end up drawing fluid inward, drawing fluid toward the proteins. And that's called osmotic pressure or colloid osmotic pressure. And so it's important to understand this diagram right here. If I gave you this equation, and could you understand what is going on? The fact that hydrostatic pressure in the capillaries is basically the blood pressure that is pushing the blood out of the capillary, and that the osmotic pressure of the interstitial fluid, so that would be any big solutes that can't pass into the capillary, that are in the interstitial fluid, are going to be drawing fluid toward the proteins in the interstitial fluid. That's osmotic pressure of the interstitial fluid. And then osmotic pressure of the capillary is going to it's going to pull fluid into the capillary because there are these large plasma proteins that are drawing the fluid inward. And those proteins cannot seep out of the capillary under normal circumstances. And then the hydrostatic pressure of the interstitial fluid, well, that's the water pressure of the fluid within the interstitium, and it's pushing against its container, moving inward. So it's important to be able to understand those different forces. If I gave you values, could you plug them into an equation that I give you that looks like this and dictate to me whether or not this is filtration or reabsorption? Filtration is when there's a net movement of fluid into the interstitial space. Reabsorption implies that there is a net movement of fluid from the interstitial space into the blood. This is reabsorption. And under normal circumstances, the arterial end of a capillary causes filtration of small solutes and fluid into the interstitial space. And then at the venous end, that blood is then reabsorbed back into the capillary. So it gets pushed out at the arterial end of the bed and reabsorbed at the venous end of the capillary bed. And since blood speed is slowed down and there is such a big increase in capillary bed surface area, it gives the tissues, the cells, the capillaries time to exchange substances. Okay. I think it's also important to have a basic understanding of the fact that, let me get back to this part. Okay. Okay. Okay, physiology of circulation. It's very important for you to understand that there are three basic ways that we can regulate blood pressure throughout the body. We can, on a short-term basis, use nerves, and that's what we do. Like if we're nervous for an exam, our sympathetic stimulation of the blood vessels increases. We have some vasoconstriction. It increases our blood pressure. Our heart is also affected. It increases our heart rate and contractility as well, but that's another chapter. So nerves can control your blood pressure on a short-term basis. Also, hormones can affect your blood pressure on a short-term basis, from moment to moment, minute to minute, hour to hour. Now, the long-term regulation of blood pressure is primarily controlled by the kidneys, which we'll cover in the third term. So, the vasomotor center, which controls the vasodilation and vasoconstriction of blood vessels, is in the same place as the cardiac centers in the medulla oblongata of the brainstem. And the vasomotor centers and cardiac centers work together. And so the vasomotor centers are either going to increase the sympathetic impulses to cause vasoconstriction or decrease the rate of impulses coming from the sympathetic nerves to cause vasodilation. And this is important because we don't use our parasympathetic nerves at all. Now, the short-term regulation using nerves occurs through neural controls, and we talked about this in class quite a bit. And we focused in on baroreceptors that detect blood pressure in the carotid sinus of the carotid arteries and in the aortic arch. That's what we focused in on, a specific reflex arc, and you should be familiar with that. Those baroreceptors end up sending impulses to the brainstem, to the cardiac centers and the vasomotor centers, and that influences how they, well, it impacts the heart and the blood vessels, and it's a negative feedback loop. Now, the higher brain centers can communicate with the medulla oblongata of the brainstem and can influence how these centers impact the heart rate, contractility, and the blood vessels. So in other words, our emotions and thoughts can affect our physical self. Now, chemoreceptors, they detect chemicals as well. We didn't talk so much about this. However, it's important to recognize that we also have receptors that detect, for example, a rise in carbon dioxide in the blood, and then that can influence the centers that impact the heart and the blood vessels. Okay, so I recommend that you closely look at this negative feedback loop when we have an increase in blood pressure in the carotid sinus and the aortic arch. It ultimately leads to a decrease in heart rate and contractility and cardiac output, and a decrease in, well, it ends up causing vasodilation, which causes a decrease in resistance and cardiac output. So initial increase in blood pressure causes a subsequent decrease in blood pressure. And then here's the other side of it. If we have an initial drop in blood pressure in the aortic arch and carotid sinuses, we ultimately impact the heart and the blood vessels to reverse that, leading to an increase in blood pressure through an increase in cardiac output and peripheral resistance to blood flow. So I would take a look at that. And also, we can, not only do we use nerves to regulate blood pressure on a short-term basis, but we can use hormones as well. And we looked briefly at some of these hormones. The only hormone that ends up causing a decrease in blood pressure is ANP, or atrial natriuretic peptide. All the other ones, like adrenaline and noradrenaline coming from the adrenal medulla, also known as epinephrine and norepinephrine. Those hormones plus angiotensin II plus antidiuretic hormone, all of those cause an increase in blood pressure except for the one that the heart secretes. It's like the heart is trying to decrease the load on itself by releasing ANP, so it doesn't have to work so hard to circulate blood. We also have long-term controls which control blood pressure, and so this is controlled by the kidney, and the kidney has two mechanisms. So one of them is the direct kidney or renal mechanism, and the other one is the indirect. So the direct renal mechanism is called that because the kidney directly, it basically does its primary function of altering the amount and the concentration of the urine in order to control blood pressure. So it directly influences blood pressure by increasing or decreasing the loss of fluids and solutes in the urine. It's the kidney doing its job. Now the indirect mechanism is that little video that we watched called the renin-angiotensin-aldosterone mechanism or system. And that is basically, that entire system leads to an ultimate increase in blood pressure. So this was the system that we looked at. Whenever we engage, when the kidney stimulates the renin-angiotensin-aldosterone mechanism, it causes an ultimate increase in blood pressure. And you'll notice that the hormones involved here include angiotensin II, which is shown as Ang2 there. It also involves aldosterone, and it involves ACE, angiotensin converting enzyme. So, if we look back at the hormones, which, did I go too far? Oh, I did. If we look back at the hormones, you can see there's angiotensin II, increases blood pressure. ADH increases blood pressure. All right. So this gives a little bit, this is a good picture to look at because it gives an overall view of the direct renal mechanism on the left that increases mean arterial pressure by essentially decreasing urine production and increasing blood volume. And then the indirect renal mechanism is shown on the right, and it shows how angiotensin II, aldosterone, and the other substances like renin all bring about this mean arterial pressure increase. Okay. This image shows essentially how mean arterial pressure is impacted by all of these different factors that if you increase peripheral resistance, you increase mean arterial pressure. If you increase cardiac output, you increase mean arterial pressure. So I think this is a good picture to look at because it basically shows a concept map of everything that we've discussed. Okay. One other thing to mention is that what is up with this thing? Hold on. Okay. All right. So tissue perfusion is basically blood flow through the body tissues and cells. The primary purpose of bringing blood through the tissue cells is intuitive. It's to deliver this oxygen and nutrients. It's to remove waste from the cells. It is In the lungs, it's a place to exchange gases. In the digestive tract, it's a place to absorb nutrients. In the kidney, we form urine from filtering the blood. So all of these things that happen because we can control blood flow. And the body is able to precisely provide the right amount of blood in order for optimal function to occur in that specific organ. So the body's pretty amazing. And hold on one moment, please. Just had to let my dog in. Okay. All right. So basically, that is pretty much what I would suggest you focus on for blood vessels. So basically, the blood vessel wall layers, places in the blood, the systemic circulation where we have the greatest pressure changes, understanding and being able to calculate mean arterial pressure, being able to give basic differences between the different types of blood vessels, the different types of arteries, veins, and capillaries, understanding capillaries and the difference between the vascular shunt and the true capillaries, and also the 3 different types of capillaries, how capillaries increase surface area of the vascular bed and decrease blood flow speed, understanding the difference between filtration and reabsorption and how all of those different forces interact. I mean, like osmotic pressure and hydrostatic pressure. Understanding what would be the impact on the body if you increased the amount of plasma proteins in the blood. That would basically cause fluid to move into the capillaries. It would cause reabsorption. On the other hand, what would happen if those plasma proteins leaked out into the interstitial space? If you had an increase in proteins in the interstitial space, would that lead to edema? Yes, because there'd be an increase in osmotic pressure in the interstitial fluid. Being able to understand the difference, the relationship between blood viscosity, blood vessel length, and peripheral resistance, or blood vessel diameter, how does that affect peripheral resistance? Understanding how peripheral resistance is related to blood flow, also known as cardiac output, and the pressure gradient. Understanding hormones that can increase or decrease mean arterial pressure. Understanding those 2 feedback loops that involve a change in blood pressure in the carotid sinuses and the aortic arch. I do suggest that you recognize that, and this is more for your future, I believe. The goal of blood pressure is essentially to keep blood pressure high enough to provide adequate tissue perfusion in order to basically provide enough blood flow through the tissues. That's the goal of tissue perfusion. That blood flow through the tissues can be regulated through local conditions in the tissues. That's called autoregulation. It's the automatic adjustment of blood flow to each tissue, to each cell, in proportion to its need. And that is controlled intrinsically at the local level simply by modifying the diameter of the local arterioles. That autoregulation is what's responsible for such great changes in blood pressure in the arterioles. There are metabolic controls, which are thought to be chemical controls, and those can regulate vasodilation and vasoconstriction at the arterioles. the metabolic or chemical controls of autoregulation are most strongly stimulated by a shortage of oxygen at the tissues. So in other words, the amount of oxygen at the cells dictates the amount of vasoconstriction and vasodilation. Also, physical factors or myogenic control mechanisms also involve the vasoconstriction and vasodilation. So in other words, myogenic control involves the localized response of vascular smooth muscle to passive stretch. So as you increase blood pressure, as you increase stretch on a blood vessel wall, that's going to affect how much it vasodilates or vasoconstricts. And then finally, over the long term, we can affect blood flow through the tissues by increasing the size of existing blood vessels and also increasing the number of blood vessels. But this happens over a long-term basis, and it's called angiogenesis. So that's important. But on a local level, at the cellular level, we can control vasodilation and vasoconstriction because there's a shortage of oxygen, or simply because we're stretching the blood vessel walls. Okay, I'd say that's the most important part of what we've discussed. And I talked a little bit about autoregulation, and that is essentially explained here. Okay. All right. So I think that's pretty much everything with respect to the blood vessels. Now, as far as the heart goes, I recommend that you understand the difference. Well, let me get out of this presentation. Okay, when we're looking at the heart, you should know the layers of the heart and the coverings of the heart. That's important. It's important for you to understand that picture at the beginning of the heart presentation that shows the outer layers of the heart as well as the coverings, the layers and the coverings. We covered most of the anatomy in lab. You understand there's a systemic and a pulmonary circulation. They circulate the same amount of blood, same volume, but just under different pressures. But as far as the coverings of the heart go, we have the fibrous pericardium, and then the parietal layer of the serous pericardium. That leads to the pericardial cavity, which includes serous fluid in that potential space. But what's important is that the epicardium is the same. That outer layer of the heart, known as the epicardium, is also considered to be a covering of the heart, which is the visceral layer of the serous pericardium. So I strongly encourage you to take a look at the picture that reviews the layers of the heart. I'm hoping you already have a pretty good handle on the anatomy and the valves and things like that. Okay. So this picture is important to look at. Okay, so this shows the coverings here. And you'll notice the visceral layer at the bottom is the same thing as the epicardium, which is the outer layer of the heart. The myocardium is the thickest wall here. It includes the actual cardiac muscle, most of which are contractile cells. And within that myocardium, that middle layer, you'll also find that cardiac skeleton. Sometimes people develop cardiac tamponade, and that is when excess fluid, it could be inflammatory fluid or blood, if it enters that pericardial cavity, it can essentially compress the heart and prevent it from being able to contract and relax. And that obviously is serious because it can lead to the heart stopping. All right. You should also know the pathway of blood through the body, beginning at the right atrium. You should also know all of the different valves and chambers and major blood vessels surrounding the heart in order to tell me the pathway of blood through the heart and the body. You should know a little bit about the microscopic anatomy of the heart, such as the fact that it has intercalated discs, which include both desmosomes and anchoring junction, and also gap junctions. And because there are gap junctions, it allows the electrical chemical current to transfer from one cell to the other, and the heart acts as a single functional unit known as a syncytium. Cardiac muscle has an abundance of mitochondria because it cannot generate ATP without oxygen. In fact, it uses all of those mitochondria to generate ATP with oxygen. So if any part of the heart muscle is starved of oxygenated blood, it begins to die. Heart muscle does not divide. Those cells do not divide, so it's not replaced with more cardiac muscle cells. It dies, loses its function, and becomes this stiff, non-contractile tissue. It's important for you to understand that the valves opening and closing and blood flow is a result of a series of volume and pressure changes that leads to blood moving through one-way valves. I encourage you to take a look at this image here, and hopefully you can look at this image and pick the lowercase letters that would indicate ventricular contraction and ventricular relaxation. Okay, so I'm going to go ahead and move forward a bit here. All right, so here are those cell junctions, the intercalated discs I was mentioning in that picture. Here you can see in the upper right the abundance of mitochondria within the the cardiac muscle cells. 99% of the cells in the heart are contractile cells, and the other 1% are the pacemaker cells that actually set the current. I think that it's important for you to understand what the different ECG waves represent. So you should know the intrinsic cardiac conduction system, how current moves from the SA node to the AV node to the AV bundle to the right and left bundle branches, and then to the Purkinje fibers or subendocardial conducting network. The primary pacemaker is the sinoatrial node because it beats at the fastest rate. We have a delay in that electrical chemical current that is almost half of the total time for the current to flow through the heart. That delay of 0.1 seconds at the AV node is important because it allows the ventricles time to fill up with blood. The only pathway from the atria to the ventricles is through the AV bundle. And the AV bundle, bundle branches, and Purkinje fibers all have an automatic rhythm that's significantly lower than the other two pacemakers, and it's about 30 beats per minute on average. The AV node's about 50 beats per minute, and the SA node is about 70 beats per minute on average, and that is with influence from the vagus nerve of the parasympathetic nervous system. Without that vagus nerve, the SA node would pace the heart at about 100 beats per minute. You should know what the, and I'll come back to this, but you should know what the different waveforms within the ECG or electrocardiogram represent. That this picture is a really good one to study for that. The P wave represents atrial depolarization. The QRS complex is ventricular depolarization and atrial repolarization. Atrial repolarization is hidden by the large amplitude of the QRS complex. And then the T wave is the ventricular repolarization. Those are electrical events, the depolarization and repolarization, and they precede the mechanical events of the cardiac cycle, which would be contraction and relaxation, or systole and diastole. Okay. It's also important for you to understand this picture, that the vagus nerve in purple is coming from the cardioinhibitory center and influences the heart rate by decreasing it. The sympathetic nerves, or fight or flight nerves, come from the cardioexcitatory center of the medulla oblongata, and they increase heart rate and contractility. Notice that the vagus nerve does not impact contractility. If you decrease nerve stimulation, sympathetic nerve stimulation to the heart, you will decrease contractility and heart rate. And so it's important to understand how this innervation of the heart affects function. Okay. Looking at this picture, it is important for you to understand what stroke volume is, how stroke volume affects cardiac output. You should know by heart how to calculate cardiac output, that it is stroke volume times heart rate, and that gives you the total amount of blood circulated throughout the body per minute. So if I gave you a stroke volume and a heart rate, could you calculate cardiac output? If I gave you an end systolic volume and an end diastolic volume, could you calculate stroke volume? Remember, end diastolic volume is the volume in the left ventricle at the end of diastole or relaxation. So that's when the heart's full, and you can see that in this picture. If you look at end diastolic volume right here and end systolic volume here, the difference between those is stroke volume. So stroke volume equals end diastolic volume minus end systolic volume, and you should know that. You should know mean arterial pressure. You should know how to calculate cardiac output by heart. You should know how to calculate stroke volume. It should just be something you know and understand. It's not necessarily memorization if you understand the meaning of end systolic volume and end diastolic volume. If there is an increase in sympathetic stimulation of the heart or a decrease in sympathetic stimulation of the heart, you should understand how that impacts heart rate and contractility. And we already talked about that. What are the factors that increase stroke volume or decrease stroke volume? Okay, so these are important This is an important equation shown in purple or blue. It's important for you to understand the three factors that affect stroke volume: preload, contractility, and afterload. Afterload is just blood pressure. Contractility is how forcefully the heart contracts, independent of heart muscle stretch. And preload affects contractility, but it's basically the degree to which the cardiac muscle cells are stretched just before they contract. So in other words, this is separate from contractility. If you increase venous return to the heart, you stretch the heart muscle cells, and then they're going to contract more forcefully. All of these affect stroke volume. So if you increase preload and contractility, you increase stroke volume. However, if you increase blood pressure, increase afterload, you decrease stroke volume. Because the heart has to overcome the blood pressure in the aorta in order to circulate blood. Okay. Yeah. So that's pretty much, those are the most important points for the cardiovascular system. When it comes to the joints, my biggest recommendation is that you study that form that I gave you. It was basically a handout that showed a table, and it was like a joint summary sheet. I think that is a good summary of the joints. In fact, if I go to, let's see if I have that under handouts. So you should understand how joints are classified structurally. That would be the cartilaginous, fibrous, and synovial joints. How are they classified? They're classified according to what material binds the bones together and whether or not a joint cavity is present. You should be able to identify the different types of cartilaginous joints and provide examples. Be able to identify the different types of fibrous joints and provide examples. You should be able to understand that cartilaginous and fibrous joints are typically classified functionally as either amphiarthrotic, which is slightly movable, or synarthrotic, which is immovable, and that all diarthrotic joints, or all synovial joints, are considered to be freely movable diarthrotic joints. If you looked at the image of, let me find my joints presentation here. There was an image I showed. There's an image I showed in this presentation of your typical synovial joint, and you should have a pretty good understanding of the structure or the anatomy of a typical synovial joint. You should know the 6 characteristics of a synovial joint as well. Sometimes synovial joints have some additional features that are not really the 6 distinguishing features of a synovial joint, but sometimes they have things like fat pads or that sort of thing. So this picture right here is something that you should know. You should understand how to label this picture and understand these 6 primary distinguishing features that all synovial joints have, and those are shown in red. Other structures that are sometimes associated with synovial joints are shown in green. You should also understand the relationship between stability and range of motion. As you increase stability, you decrease range of motion of a joint and vice versa. You should, oops. My iPad is kind of freezing here. But look at all the different types of synovial joints. We have a picture that shows the different types of synovial joints. And you definitely ought to take a look at that. Things like the plane joint, the condylar joint, the saddle joint, the pivot joint, the ball and socket. That's what I mean. Understand whether or not they're non-axial, uniaxial, biaxial, or multi-axial, and what the basic shapes look like. Also, you should have a pretty good understanding of movements. So if I said, you did a jumping jack, what are the movements involved? That would be abduction and adduction. And what plane would it be in? So anyway, that's what I would focus on for joints. And then for blood, you should know the basic functions of blood. What type of principal tissue is it? There are 4 principal tissues. There's epithelial tissue, connective tissue, nervous tissue, and muscle tissue. Those are the 4 principal tissues. What is blood? What's the difference between the buffy coat blood plasma and the erythrocytes that make up hematocrit? Why is blood plasma considered a connective tissue? Why? What is the blood plasma? What are the fibers? What's the value of plasma proteins? What do they do? What are their functions? What are the functions of plasma proteins? The most important or abundant plasma protein is albumin. What does it do for the body? It does a few different things. It buffers blood. It contributes to colloid osmotic pressure. It carries other molecules that are insoluble in water. Why is the erythrocyte such a good example of how form affects function, or complementarity of form and function? What are the reasons for that? What are the different types of leukocytes or white blood cells, and what is their primary function of each of them? How is blood formed? I mean, what cell is the major stem cell that can differentiate into every type of blood cell? What cell makes blood cells? And what hormone? Think about that essay question we did. Every little practice essay question we did is somehow in this exam, either as a multiple choice question. And we talked about the hormone that stimulates the production of erythrocytes, how kidneys secrete erythropoietin, the interaction of the red bone marrow of the skeleton, the blood of the cardiovascular system, and the hormone of the kidney, and how they all interact with one another. So look back at those practice essay questions and know those. And that basically concludes my study guide. That's about an hour long. I'm going to go ahead and stop here. If you have questions, email me.